Hi, folks. Welcome in. Hi, everyone. Hi, everybody. Good afternoon, everyone. That was so loud. Was it? Oh, that's interesting. Hi, Kiora. Hi, Joseph. Hi, Natalia. I was chilling with music, and then I ruined it. Well, don't you worry. Yeah, we're doing pedigree charts today, Maddie. That's our goal for the day. I'm happy to ruin that for you, Daniel. Am I? Why am I so loud? Hold on. Maybe am I louder than normal? My internet is super slow. No. Why is my internet super slow? How do you know it's super slow? Me confused. How do you know it's so slow? Because of the way that I sound? Does it sound like my voice is chugging? Where's my VPN? Maybe it's because my... Let me check. Also, it was in... Why was that meeting so bad? Because your YouTube's been loading for so long. Oh, why was the meeting so bad? Also, it was in yesterday's homework, so like, why would it be a sign if we're not doing it today? That's weird. It shouldn't have been yesterday's homework. For me, everything's normal. My internet's been bad too. Yeah, let me see what I gotta see what's going on. It shouldn't have been in the homework questions from the textbook. Oh, okay. Well, I think I maybe made a mistake on that one. That's okay. We cover them today. So worst case scenario, we'll cover that stuff today, and you can just skip those. Hopefully, you skipped those questions yesterday. Good. Perfect. That's weird. Maybe I think maybe that homework was supposed to be assigned today. Anyways, regardless, it doesn't matter. It's done is done. So far, you're the only person that's asked about them. So happy days, happy days. Alrighty. It's weird that I'm so loud today. Or at least maybe it's just to some people. Maybe you just normally have the volume turned down and today you have it turned up. Who knows? All right, let's take a look at our attendance. I have your schedule for today up and rolling. And we'll go from there. Wait, can you attach files in this chat? Yes. Like from Google Drive, etc., etc. Why? No, like you are so loud, my volume is like on 5. That's weird. Hey, Ken, how's it going? Yeah, it's pretty neat. Weird, I have no idea what to tell you on that one, Maddie. Alrighty, let's take a look at attendance and we'll get started in just a minute or so. Attendance here, we've got 28 folks. Who are we missing? First name sort. Christina. Yeah, we're going to talk about all of that stuff today. There are people cleaning my kitchen, but I'm hungry. Oh, dear. Let me just finish taking attendance real quick. Megan, Maddie, Luca, Kiora, Ken, Joseph, Jonathan, Janice, Jaden, Helen, is your YouTube screen frozen? There. Is that better? Yeah, I don't know why that's when someone was like your YouTube screen, I was like, what YouTube screen? I don't have YouTube open. Helen, Han, Hamid, Emily, Dominic, Daniel, Celestine, Christina, Catherine, Ashley, Arlena, Anna, Alicia. What are we missing? Okay, I'm missing someone and it's going to bother me because I don't know who it is. Vanessa, Tammy, Natalia. Ah, she thought I found her. All right, sweet. Sweet. Let's get started. Bad teacher, you don't know who you're missing. I know. I know. If I could see all your faces, it would make my life easier, but I digress. Alrighty, so there's a couple of things I want to talk to you about today. The first, we're going to look at tracking inheritance. So that's awesome. That's our lesson number 7 that we'll be looking at today. But I also wanted to show you, because a couple of people have been asking about the homework to take up. And so with regards to those worksheets, the textbook questions, I'll always leave those to kind of your devices, just because the answers in the back of the textbook don't always match up. They are there if you want to look in the appendix. It's just they don't always match up, slash, or sometimes even wrong, but I digress on that one. The big thing I wanted to share with you is from lesson 6, at the end of that worksheet, at the end of the lesson, there was a practice problem or a bunch of practice problems. I have posted the answers in here, practice problems, questions, and practice problems, answers. So if you open up this guy, you'll see all of the answers to those questions for you to check your work against, okay? So I would say before I take up any of those, double check your answers to that file. And then if you're like, I still don't know where I made a mistake, I'll be happy to talk about, oh wait, hold on, you can't even see this. Good Lord, sorry folks. I'll be happy to, so in our Unit 2 folder, genetics practice worksheets and answers, open that fella up. And lesson 6 practice questions extra are there with answers, and then the practice problem. So all of these answers from the worksheet from lesson 6 are there. If you got them all right, excellent. If you had a question or wanted me to take up a specific question, I'll be happy to, but I'm not going to take up every one of them. All right, that's too much time. We don't have time for that. So if you have a specific question, I'll be happy to answer, and I'll happy to take it up. But if you're not sure where you went wrong, check your answers, see if you can figure that out. And then if you still have a question about it, I'll be happy to take it up. So it's in the Unit 2 folder in our genetics practice worksheets and answers under the lesson 6 answers, okay? Any specific questions about that, or does that make sense? Yes, the worksheet from the in the lesson yesterday. Welcome back, Emily. Alrighty. So today is lesson 7. Let me just get it sorted. If those of you who have questions about the homework, if it's a specific question, I'm happy to answer. But if you need to take a look at all of those before you ask a general question, and then if you can't figure it out from the answer key, again, I'm happy to take it up. Okay? Because I know 1 or 2 people were asking about the homework answers, and I was like, okay, I'll just post it because more than one person asked. So there you go. You have all of them, access to all of them. A lot of practice. I will say for this lesson or this unit specifically, it's like math. The more you practice, the better you will be at it. So I've given you an absolute metric ton of practice. The more you do, the better you will be at it, straight up. So let's get started with our lesson today. Oh, well, is there any questions regarding that homework specifically, a specific question? I'm going to take up the last question or a question like it from that worksheet homework. So good? Okay. Alrighty, let's get to it, folks. Lesson 7. So lesson 7 has us tracking inheritance, Now, there will be questions. You saw a practice question like this in your worksheet yesterday. They will come up consistently where we will need to work backwards. Okay? Where we will need to work backwards. And the way we work backwards is to look at the offspring outcomes and then make predictions about the parents that we see. So in this question, we have a cross between 2 chinchillas, and 2,020 of them are gray, whereas 675 are white. And we are asked to determine the genotypes of the parents. Which allele is dominant? Assume the trait is influenced by only 2 alleles and follows the laws of Mendelian inheritance. We will use capital R and lowercase r to represent those alleles. So first thing, we have to let capital R represent gray and lowercase r represent white. Now, why would we do that? Why would we do that? Well, because we use the offspring numbers to determine which one shows up more frequently. And again, right off the bat, you should be able to look at this and say, wow, there's almost a 3-to-1 ratio of gray to white. So that trait is expressed in the population more. Therefore, we would expect it to be dominant. And we can do some quick arithmetic to determine that. So when we add the total, 2020 plus 675, it gives us 2695 in total. That's how many offspring we have. Of those total offspring, 2020 are gray, which is approximately 74.9%, which means that approximately 25.1% are going to be white. Oh, I didn't erase that part. There we go. 25.1% are going to be white. And as a result of that arithmetic, we can see that a 3-to-1 ratio exists in the offspring of gray, 3, to white, 1. So we've determined that that gray is the dominant trait. And now we can look at the potential outcomes, of the, or what would lead to that potential outcome, I should say. So we have to consider the gray being dominant, white being recessive. And when we pretend, and again, this is like not pretend, but when we work backwards and we show a cross, what would yield that outcome? Okay, we need to figure out what would yield that outcome. And now we're already pretty good, I would say. We're already pretty good with our understanding. If there's a 3-to-1 ratio, we know automatically that it has to be 2 heterozygous parents. But what if we didn't know? What if we didn't know this? How would we solve this? What if we got different values? How would we solve this? Well, the way I worked at it is I know that there's approximately a 3-to-1 ratio of gray to white. So I started thinking of it like this. So I have to have 3 capital R's at least, because it at least has 1 capital R, right? It at least has 1 dominant allele if we're going to see that 3-to-1 ratio. And the only way that we get that white allele expressed is if we have homozygous recessive. So right off the bat, that's how I start. Okay, so now I have an incomplete genotype of each offspring. Well, I know that because this is homozygous recessive, that in order for this Punnett square to be true, let me just add the top parts up here. In order for this Punnett square to be true, I have to be able to have a parental genotype that donates that lowercase r from both parents. So I know that one of the alleles here has to be an R, and I know that one of the alleles here also has to be a lowercase r. So now I can finish distributing it into the remainder of those rows and columns. This is a little janky, but you'll make it look much nicer than mine. And we're good there, okay? And we're good there. Now, because this recessive allele came from here, we know that this dominant allele comes from here. Does it matter how you organize the R? No, it does not. So because we know that that capital R, or that dominant allele, came from there, we have that. And so that takes care of this one. That takes care of this one. Let me highlight it. Oops. That takes care of this one. But then what about, let's use burgundy. What about this fellow? Where did this guy come from? Well, we know it has to come from up top here. And if that's the case, we must have that dominant allele as that allele up there. And again, when you take a look at each of the alleles that are present, we have, as a result of that working backwards, we have 2 heterozygous parents. And that's what we started off with at the top. And we know, again, we know this because that 3-to-1 ratio only comes up when we have 2 heterozygous parents made. Okay. Questions? Questions. Nope, sounds good. Alrighty. Now I'll preface this one right off the bat for this next part. Maddie, you're going to be very excited. You're going to be very excited. Get ready, Maddie. I told my period 1 class that I have a student in my period 3 class that has rabbits and that they will be stoked to see this practice problem question. And they all thought that it was very nice that you would be very happy and excited for this question. So from my period 1 class to this class, they say hello. They say, I hope you enjoy the rest of the lesson. My kids in period 1, like I said, all my kids, all of you, every single one of my sections, you're all great. My stinky babies, long and short. Yeah, exactly. All right. So if there are no questions, let's extend the idea. Let's extend the idea of working backwards into some theoretical concepts. And those theoretical concepts are what's called a test cross. Now, I'll preface this right off the bat. A test cross is not always going to give you results that you expect, okay? This is where the probabilistic nature of genetics comes into play, because in every single one of our examples so far, we've had clean-cut data, 75% red, 25% white, 75% dominant, 25% recessive. It doesn't always work that way, okay? Those are just odds, all right? There's variation and variance, it's called, within that range, okay? And in order to determine unknown individuals' genotypes, we perform what's called a test cross. So a test cross is used to determine whether an individual that displays the dominant phenotype, we're trying to figure out if they are heterozygous or homozygous. So in the example of our chinchillas above, we're trying to figure out if a gray chinchilla is homozygous dominant or heterozygous. Now, in order to do this, we must cross the one parent with unknown genotype with an individual who is homozygous recessive. This is the crucial one. This is the crucial one, sorry. It needs to be homozygous recessive. So one parent is either going to be homozygous dominant or heterozygous, or the other parent is going to be that homozygous recessive. We always have to have a homozygous recessive parent in a test cross. Always, 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 okay? Always, always, always. Does everyone understand that statement? This is crucial to our understanding of test crosses. We must have a homozygous recessive parent that's involved. Must. And the reason being is that they are a known genotype. It's a known genotype because the unknown parent, which could be homozygous dominant or heterozygous, we don't know. We don't know. But in order to be that recessive trait, that recessive phenotype, you must be homozygous recessive. There's no other option for it. And we can use this understanding to do a cross to determine how the offspring should shake out and what it would look like based on all of this information that we are talking about here. Okay, so let me just scroll down a second, and then we'll talk a little bit about this question specifically. Are there any questions about test cross? General questions about a test cross? General questions about a test cross? General questions about a test cross? So wait, I'm waiting. No rush. And I might also explain your question, Joseph, in the example. So happy to answer it. If it is a question, one parent can be homozygous dominant or heterozygous dominant. Yes, if they display that dominant trait. Right? Yeah, so I'll show you in this question. And this is the question that I know Maddie's going to be stoked for. So here we have rabbits. In rabbits, long ears are dominant to short ears. Long ears are dominant to short ears. We are performing a test cross. This is inherent in this procedure that I'll show you coming up. It's performed on a rabbit with long ears. So we have a test cross performed with a rabbit that has long ears, and it results in 3 rabbits with long ears, 1 rabbit with short ears. Determine the genotype of the long-eared parent. Please use a Punnett square to explain your answer. White bunny's ears are longer than brown bunny's ears, so the long is RR or no. Well, hold on. We don't know, right? That's the trick. That's the trick of it all. We don't know. But we'll let E, if you used R, that's fine. We'll let E represent long ears, and we'll let lowercase e represent short ears, okay? So capital E represents long ears, lowercase e represents short ears. If you want to use R or E or L, I don't care. It doesn't matter at the end of the day. All that matters is we have our representative of our dominant allele and our representative of our recessive allele. Now, the one long-eared parent, we do not know if they are homozygous dominant or heterozygous. We have to test for that. It could be either of those, either dominant or homozygous dominant or heterozygous, okay? We don't know. So we have to perform 2 tests for each of these. So allele number 1, we're going to test allele number 1. Actually, I'll put a box. Allele number 1. Bam. Allele number 1. So we perform a test cross. And remember, we perform a test cross with that homozygous recessive parent as the other option. Because again, it's a known phenotype and a known genotype. Now, take a look at this Punnett square. What do you notice? What do you notice? Yeah, all the genotypes are that. What does that give us? Good, Maddie, that's also correct. 100% what? 100% what? 100% long ears. Is it possible to have any short-eared offspring like we have in our example here? Is it possible to have any short-eared offspring? It's impossible. It's never going to happen unless a disjunction disorder happens. But even then, that won't actually yield to it. So it's not possible to have that short-eared offspring. So can we confidently say that the parent is not going to be homozygous dominant? Yes, exactly, Joseph. As long as there is one dominant allele, it's impossible to have that recessive allele be displayed as a trait. So as Maddie says, it's impossible to have that recessive allele displayed, and therefore we can absolutely get rid of this as an option. It will not be homozygous dominant. All right, option 2. Option 2. What do you notice? What do you notice about the Punnett square? What do you notice? Okay, careful now, Maddie. Careful. Yes, it does not coincide with what we see in the offspring. But recall, a Punnett square just gives us a probabilistic outcome. It doesn't say that every 4 children, half will be long-eared, half will be short-eared. That's not how it works. It's just a 50-50 chance. It's just a 50-50 chance that they will be long-eared and short-eared. In reality, you could still end up with 3 rabbits with long ears and 1 with short ears, just because of all of that random assortment, all of the crossing over, all of the random fertilization. It's still probable or possible to have that outcome. I didn't say I was wrong. It doesn't coincide with what we have. Yes, but we're not looking for it to perfectly coincide. And this is the cool thing about these test crosses. We're not looking for it to naturally coincide. All we want is to have the potential to have short ears. And we get that. We get that with option 2. So option 2 is a yay. The parent's genotype is most likely heterozygous, because this is the only way that we can get short-eared offspring. Alrighty, questions about this test cross? The white one had broken ears. Oh dear, it doesn't stick up like it should. He looks like an L. Alrighty, does that test cross make sense to everyone? There are no specific questions, or? Right, sweet. Check the, okay, I got the check marks from some people. Thank you so much for those check marks. Happy to see that it makes sense. All right. No questions, eh? No questions. All right, let's move on. So we will take a look at pedigrees next. And that's my apologies. I didn't realize that I assigned pedigree homework. That wasn't supposed to be the case. I think what ended up happening is I assigned today's homework in the textbook to yesterday or to lesson 6. My bad, folks. My bad. All right, let's take a look at pedigrees and pedigree charts, because now we can put together all of our understanding of phenotype, genotype, homozygous, heterozygous, test cross, Punnett squares, probability outcomes. All of that can now be attributed and associated with the tracking of inheritance over what's called pedigree charts. And we can track that inheritance over many, many generations. Over many generations. How many Marina's doing? Oh no, we're good. Okay. I can track that inheritance over many generations. Over many generations. So what do pedigree charts tell us? Well, it shows us biological gender. It shows us relationships between people. Oops, there we go. It shows us relationships between people, and it shows us the phenotype for a given trait. So a lot of information can be had in these pedigree charts. Okay, a lot of information can be had in these pedigree charts. And here we have the legend of these pedigree charts here. Okay, this legend, I have students that like straight up copy and paste it into their notes, or they write it down and they hold on to it and utilize this information, because it will be important from now until the end of this unit. And I'll even bring it back. I'll even bring it back in our evolution unit as well, because it is important to understand what all of these things mean. Okay, it's important to understand what all of these things mean. So we have male, which is a square, female, which is a circle. Affected male is the, oops, let me just, there we go. Affected male is a filled in square. Affected female, a filled in circle. And when I say affected, I mean it has the trait. It has both of the dominant alleles, or has at least one dominant allele. It has the condition that we're talking about, what have you. That is affected. When I say affected, I mean all of that. Affected is also the dominant trait. And then we have the joining of mates, signified by a dash that connects to a square and a circle. We have siblings that are connected by a joining T. Identical twins, which is a triangle within a triangle. And then fraternal twins, which is a triangle with its base up on top. I'm able to go through this again briefly because you cut out. I think it was just my internet. Yeah, so the key thing here with regards to these is that you understand, the big one to understand is this, affected versus unaffected. Oops. If you can get those, the rest will fall into place. Affected means it has that trait, the trait we're talking about in question, or it is the condition that we're looking for. Okay, it's the condition that we're looking for. That's the main thing. If that cut out for you, Dominic, that's the main thing that you should have. Cool. All righty. Now, let's take a look at each of these components, because here on the left, we have a pedigree chart that has 3 different generations. So the Roman numerals represent the generations over time. 1 being the oldest, 3 being the most recent. My attendance is sent in. Beauty. Okay. Yeah. So 1 being the oldest, 3 being the most recent. The Arabic or the Indian numbers, as one of my students was like, corrected me earlier today. These are the 1 to 4. 1 representing the general birth order. So 1 being the oldest, 4 being the youngest. Or in this example, 1 being the oldest, 3 being the youngest. In the example where they are offspring and siblings. And then the last thing is that we can use these numbers to give specific titles to people. So I can say 2-3, and that will refer to the 3rd male in generation 2. Or 2-4, which is the spouse of 2-3, or the mating partner of 2-3. And this allows for us to make to draw specific reference to specific individuals within that pedigree chart, to allow for us to compare, contrast, and overall just look at the general ideas of each of the people that we're dealing with. All right, questions with regards to the pedigree chart, or at least the introductory pedigree chart. All righty, no questions, it seems. Are we gonna get the problem set today? Yes, I will assign it along with a video that kind of talks a little bit about it at the end of the day. And then you can look at it for homework if you would like to. Alrighty, let's take a look at why this is useful. Because the usefulness of a pedigree chart, if you do choose to pursue a degree in biology or something specifics with regards to genetics, you could potentially get into a career as a genetic counselor. And they use these pedigree charts to help trace the genotypes and phenotypes in a specific family. They also help to identify whether a particular trait runs in a family and what that might mean for those future generations and offspring. It's also used by livestock geneticists as well as farmers to kind of track and to ensure that specific livestock traits are conserved, as well as agricultural, as well as breeders. So there's many, many, many different ways with which we can utilize these wow, pedigree charts. I just had a brain fart there. These pedigree charts. Okay. So we're going to look at a specific example coming up. And again, we will tie in all of these ideas. All of these ideas will be tied in to some of the general concepts that we've looked at to date, because there's many, 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 many ways that they can be connected. But I will just look at it generally today, and we'll start getting into the thickets of it as we move through the rest of this unit and the lessons both today and tomorrow. All right. So let's take a look at this example. Here we have a very, very long-winded worded question. And this is just how they're going to look. Because written down, the information is not as concise, and it's quite a bit. So in our example, we have a family that we're going to look at, and we're going to create a pedigree around that family. And that family starts with a brown-eyed woman. A brown-eyed woman whose father had blue eyes, and whose mother had brown eyes, So whose father had blue eyes and whose mother had brown eyes. This brown-eyed woman marries a brown-eyed man. The father's mother or the mom's mom and dad. So the mom, the first mother that we're talking about, the brown-eyed woman, has a blue-eyed father and a brown-eyed mother. That brown-eyed woman marries a brown-eyed man whose parents were both blue-eyed. Holy. You'll see as we tease it out. And then finally, they have a son who is blue-eyed. Draw a pedigree, assuming brown eyes are dominant to blue eyes, and then we'll include genotypes later. So she remarries. No. So here, let's start before we try to like figure out all of the drama that's in this family. Wait, but it says whose parents are also brown-eyed. Yeah, that's a misnomer. A brown-eyed woman whose father had blue eyes and whose mother had brown eyes. Okay. She marries a brown-eyed man whose parents are also brown-eyed. Oh, I highlighted that wrong. Whoopsies. I highlighted that wrong. Maybe that was a source of confusion. His parents are also brown-eyed. Woman one has a mom with brown and dad with blue. Correct. Woman one marries brown-eyed man and his parents have brown eyes. Correct. The spouse of the woman's parents have brown eyes. Correct. Woman one and man one have blue-eyed kid. Correct. So let's take a look. First of all, my brain is dying. I know. That's why the pedigree charts really help, but we have to tease it out. First of all, oops, too early. We have our generation and we have our legend here, okay? We have our generation and we have our legend here. This is crucial to help us understand who we start with and where we need to go. So the legend, brown-eyed male, brown-eyed female, because again, we said just like in the last sentence, capital B will represent brown eyes, which is the dominant trait or affected. And then lowercase b is blue eyes or unaffected. This is hard to create on a computer. Yes, I agree. This is why I set it up this way, but it's not perfect. It's not perfect. All right, so let's take a look. First of all, I named my pedigree chart, tracking eye color in a family. Tracking eye color in a family. I have my legend, brown-eyed male, brown-eyed female, as we are talking about it in the affected versus unaffected way. So the brown-eyed males and females are filled in. Blue-eyed males are not filled in. We have a first, second, and third generation because we talk about a woman, her parents, and then her kid. So 3 generations in total. And we start with the brown-eyed woman, who I have or I've shown you here. So this brown-eyed woman has parents who are blue-eyed and brown-eyed. Dad is blue-eyed, mom is brown-eyed. With me so far? So this is, I'll highlight it just to kind of, this is this family here that I've shown you. A brown-eyed woman whose father has blue eyes and mother has brown eyes. That's what I've shown you here. Okay, she marries a man with brown eyes. Ooh, spoiler alert. A man with brown eyes. Come on now. Oh, God. A man with brown eyes. No, that's why. There we go. A man with brown eyes. There he is. There he is. A man with brown eyes. His parents are also brown-eyed. His parents are also brown-eyed. He must be shy. So here we have the whole family. And then finally, we have the last piece of the puzzle. The last piece of the puzzle. Those parents in generation 2, they have a child with blue eyes. With blue eyes. All righty. We've set up this pedigree chart. Start to finish. Start to finish. We have our grandparents, we have the parents, and we have the child. Cool, I know. So that's part 1. Set up. Much easier to see in this pedigree chart than reading it in that mishmash of language. Wait, so how do we know to choose between affected and normal? Affected will be the dominant trait always. Affected will be the dominant trait. Yeah. Unaffected is recessive. That's just, they're synonyms for each other. Alrighty. So part 2, I would like us to determine the genotype of each individual. And I have an asterisk here. We have capital B underscore. It means either homozygous dominant or heterozygous is possible because we don't know unless we perform a test cross. Now, the easy ones, if they are blue-eyed, bam, bam, no problem, right? Because it's that recessive trait. The only way we see that recessive trait is if the genotype is homozygous recessive. So the blue-eyed people, no problem. EZ, PZ. Lowercase b, or homozygous recessive for both of them. Now, where things get dicey. Do we know the genotype of the brown-eyed mother? I1, generation 1. Do we know the genotype of that mother, the grandmother on the left? No. Do we know that she has a dominant trait though? Yes, because she's brown-eyed. So we signify it by saying B underscore. That second allele, it could be the recessive, it could be the dominant. Because the father is already giving one recessive allele to its daughter, spoiler alert. So we know that that recessive allele is going to come back potentially later on. What about the other grandparents? Do we know what their genotype is? The 1, 3, and 1, 4 grandparents. Do we know what their genotype is? They have brown eyes, but do we know what their genotype is? Yeah, we don't know definitively. Maybe Kiora, but which one? Which one would be heterozygous? We don't know. We don't know. It's impossible to determine unless you did specific genetic testing, but it's impossible to determine otherwise, right? So we know for sure that they're brown-eyed, so they have to have one dominant allele, but we don't know what their actual genotype is. Okay, beauty. What about generation 2? Can there be 3B? What do you mean 3B? No. Remember, these are they're homologous chromosomes, one or the other. There's only going to be two. There's only going to be two. Yeah. Okay, what about generation 2? What about generation 2? We know that they are brown-eyed, so they have to have that dominant allele. But they have an offspring that is blue-eyed. So both must be heterozygous in order to have a chance, at passing on that recessive allele that gives their child blue eyes. Now, for those of you following along from the last 2 lessons, this is a fancy way of saying P generation, F1, and F2. And here we have Mendel's inheritance coming up once again. We would expect that a 75% chance of being brown-eyed, and we would expect it to be only a 25% chance of being blue-eyed. But here we have this little boy that's given us hope of a small chance of blue eyes, and so it is. Mendel's so annoying. So now the brown will never pass on through this child. Correct. Through this child. They might have another child with brown eyes, and then it could be passed on. But for this child, it's impossible for this child to pass on a gene or to give brown eyes to its offspring. It will never happen. That is so cool. I agree, Kiora. This is the beauty of genetics as we start to make it connections to the overall ability of our understanding of how actual genes are passed on. Loser. What a loser. He can't even pass on brown eyes. What a loser. Anna, so kind and compassionate. All righty. So that's how a pedigree chart works. So now my hope is that those of you who took a look at those homework questions and were like, what the hell is this guy talking about? Hopefully it makes a little bit more sense to you now. All righty. Any general questions about pedigree charts? Is that why blue-eyed people have a common ancestor? Yes and no. Yes and no. Like all traits had to have happened as a result of one specific mutation. And then that mutation gets propagated down throughout the rest of those pedigree charts. So technically, yes, they would, but it'd be like their 11,000th cousin or whatever. Incest. Well, no, not necessarily. All righty, let's move on to the sex-linked inheritance components, where we will start to connect our understanding of pedigree charts, Mendel's genetics, and expanding all that and making those sex-linked inheritance connections. Imagine an alternate universe that blue is the common color. Yeah, well, all it takes is for one random mutation to happen one way or the other, and different traits could be different dominants, right? It could have different dominants. Alrighty, so what is sex-linked inheritance? Well, it follows the X and Y chromosome, i.e. the sex chromosomes. Because recall, the X chromosome is unique. It is not homologous. It does not have a homologous pair all the time. Because in males and females, even though they have those similar 1 to 22 chromosomes, their 23rd pair is not homologous because a common biological female will have 2 X chromosomes, so 2 of the X chromosomes, whereas the common biological male will only have 1 X chromosome. So for issues or traits or diseases that are on the X or Y chromosome, you will only see it pop up in one specific gender as a result of many different factors. So if an allele is found on any of the other 22 chromosomes, we consider that an autosome or an autosomal inheritance. Everything we've looked at till now has been autosomal. Okay, everything we've looked at now has been autosomal. Now we will start to connect the sex-linked components. Isn't color blindness only in men? Technically, yes, but also not technically. I'll talk about that later. We'll have examples with color blindness. All right. So if an allele is found on the X and Y chromosome, then we consider that sex-linked inheritance. So sex-linked inheritance means that the allele for the specific trait is on X or Y. These disorders can be either X-linked, which is a phenotypic expression on an allele that is found on the X chromosome, like color blindness, as Kira suggested. Hemophilia A, which is a blood disorder, a blood clotting disorder, and male pattern baldness. Those are all X-linked. And we describe X-linked as X to the exponent of lowercase r or X to the exponent of capital R. What does R represent? That could be any trait. It doesn't necessarily matter. I'll use it in a specific example later, but it could be any trait. The recessive or the dominant is still denoted by a lowercase or a capital letter. Yeah, it could just as easily be an A. It could just as easily be a C, whatever. All right, but then we can also have Y-linked. And recall, because the Y chromosome is smaller than the X chromosome, even though there are phenotypic expressions of the allele that are found on the Y chromosome, like reduced fertility in males, there are far fewer genetic disorders associated with the Y chromosome. Again, because it is male specific, but also the amount of DNA is significantly smaller than those of the X chromosome. And we denote any X or Y-linked allele with Y to the exponent of capital R. Now, why would we describe this? Why would we describe this as Y to the capital R only? There's no lowercase r here. Why would we describe it as capital R only? Let's see if someone can guess or figure it out. Let's see if someone can figure it out. I'll give you all time to think about it. Let's see if you can figure it out. Because there's no homologous pair. Ooh, dominant, because Y is recessive. Good guess, Emily. Does it have to do with you can only have XY, not YY? Yes, Kiora, and that's based on what Dominic said. That's okay, Emily. Good guess. So Dominic and Kira, your answers are both, you're both connecting your answers there. There's no homologous pair for the Y chromosome. So the trait will either be there or it will be non-existent. So you don't have to worry about stating a recessive allele because there technically is no recessive allele. Recessive implies that when in the presence of another allele, if it's also recessive, that trait will be shown. If it's in the presence of a dominant allele, that recessive allele will be suppressed. But because there's only one Y chromosome, it's either going to have it or it won't. There's no dominant or recessive. So Y is dominant because there's no recessive. Yes and no, Kiara, kind of. And I'll show you in the next example, okay? I'll show you in the next example. All right, let's take a look. So sex-linked inheritance. Hemophilia A is an X-linked recessive disorder. Holy, now we're starting to speak some science here. X-linked recessive disorder. X-linked recessive disorder. Predict the probable outcomes for the cross between a mother who is a carrier. Mother who is a carrier of hemophilia. So mother is a carrier of hemophilia. And the father does not have hemophilia. The father does not have hemophilia. All right, so what does it mean to be a carrier? What does it mean to be a carrier? So let's just take a quick look here. You have it in your genes, but you don't suffer. Yes, so I will tell you right off the bat, carrier is equal to heterozygous. Carrier is equal to heterozygous. You have a trait in there, but it is not expressed, okay? So we will use our nomenclature. Oopsie, spoilers. We will use our nomenclature to describe, what is that? There we go. To describe what each trait will be represented by. So we let X lowercase r represent hemophilia because it's an X-linked recessive disorder. And X capital R will represent the unaffected, Okay, so X lowercase r recessive represents hemophilia. X to the exponent of uppercase R is the unaffected, So because we know the genotypes of the parents based on the statements, we can state that the mother is a carrier, so is heterozygous, if you will, heterozygous. The father is not affected by hemophilia, so it would only have the one single dominant allele. With me so far? Good. Okay, what are you confused about? That's okay. This is new stuff. It's tricky. I just want to eat food. Why are we doing this right now? I know. We're almost done. This is the last question I'm going to talk about today, and then I'm going to release you to go eat food and relax for a bit. And I'll take up the rest of it tomorrow. All right, let me finish taking up the question, and then let's see if that alleviates some confusion. Okay, so we are going to do probable outcomes for a cross between the mother and father. If the carrier is heterozygous, which we talked about already, what will that cross look like? And here we go. So we have the mother who is a carrier up top. The dad who is unaffected here. And then they each give one of their chromosomes to their offspring, right? Just like in the autosomal inheritance, it's no different, okay? So we have 4 outcomes. The first outcome, 1/4 of the outcomes, the female is unaffected. Why is she unaffected? Well, she does not have 2 recessive alleles, and And she doesn't even have one recessive allele. So we can't even call her a carrier. She is not a carrier. Okay? So one is a female unaffected. One is a female carrier. Female carrier. She has a recessive allele, but also has the dominant allele. So she will be unaffected, but she is a carrier. Her offspring potentially have a chance to get hemophilia. Next, we have a male. One of the four males is completely unaffected. One of the four males is completely unaffected. He has one dominant allele. That's all he needs. No problemo. No problemo. And then lastly, unfortunately, we have one male who is affected. They have the recessive allele. One recessive allele is enough to give a male hemophilia because the Y chromosome carries no genetic connection to the hemophiliac trait. Four distinct and different outcomes based on our deduction of the parents' genotypes. All right, questions. Specific questions. If you're confused, that's okay. Maybe you need to think about it and digest it. But specific questions. So a woman needs to have needs to be affected, and a man only needs one. Yeah, so a woman needs two recessive alleles to be affected by X-linked disorders, recessive disorders. A man just needs one of those X-linked recessive alleles to be affected. Correct, Yora. Correctamundo. Do all the Y's still have to be superscript R? No, there's no associated superscript with the Y's. Only X's in this example. Because it's an X-linked recessive disorder. Right? Right at the beginning of that question, it really sets the stage for what kind of expression we're expecting for in this question. Right? That's why I'll tell you right now, folks, those of you who take your sweet time reading these questions and really try to parse it out, you will have the most success. Those of you who try to rush through it, it's going to be tricky. And like anything in mathematics and science that requires practice, this is one of those things. The more you practice, the better you be. The recessive is x to the exponent of lowercase r. Correct. The dominant is x to the exponent of uppercase r. Correct. Are we doing the rest of the examples tomorrow or homework? Tomorrow. We'll do them tomorrow, Anna. I'll take them up before we do our next lesson. Because these are important. The sex-linked ones are the hardest thing you'll probably do in this class. Just because the pattern isn't quite evident right off the bat. Okay. All right, folks, there's about 10 minutes left of class. I'll release you. I won't do any more examples because I think your brains are full, so to speak. So go take a 10-minute breather before your next class. We will take up the rest of the examples tomorrow. Okay, folks? Have a good one. Hopefully you found some useful information. Try the practice questions in that folder, and I'll see you all online tomorrow, folks. Bye, everyone. Yeah, we'll do the rest of the pedigree chart. I'll do some more tomorrow. Don't worry. We'll do the rest tomorrow. I just wanted to cover a little bit of each. Yeah. See you later, folks. Bye.